Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, as you know that we are carrying a series on plant physiology, and so far we have conducted numerous sessions, numerous lectures. In our previous session we talked about plant hormones. We would be continuing our lecture further and we would be talking on plant hormones today also, and for this discussion we have again with us in our studios Dr. Ashish Kumar Nandi. Dr. Ashish Kumar Nandi is a professor in School of Life Sciences, Jawaharlal Nehru University. So dear friends, let's welcome our guest Dr. Ashish Kumar Nandi, and let's understand about plant hormones under his able guidance. So first of all, I would like to welcome our guest Dr. Ashish Kumar Nandi. Dr. Nandi, welcome to the research lecture. We discussed about plant hormones in our previous session, in our previous lecture. Today also we would be talking more on plant hormones.
Right.
So over to you, sir. We are curious to know what are we going to learn in this session today through you.
Okay, thank you, Geetika. So in the previous class, I have talked about different kind of hormones which are there in the plant, and also I have talked about what we call as phytohormones and how they are different from or similar with plant or animal hormones. And I also briefly discussed about the presence of different types of plant hormones, like 5 classical plant hormones, which include auxin, cytokinin, gibberellins, abscisic acid, and in addition to ethylene, and in addition the newly discovered hormones like brasinosteroid, strigalactones and so on. And also I have in quite a good extent I have discovered how auxins was identified in very early research in late 1800s. And I also have discussed, that is the main thing, how auxin was discovered. So to give a very brief about that, I would like to tell you today, so that most of the students, if some of you have not listened to my last talk, you should be able to follow it. The Auxin effect was first identified by looking at its role in photomediated seedling growth, something which is observed by most of you. If light is falling in one side of the plant, then the plants bend towards the light. So that is what is called as phototropism. And from the basis of the phototropism, the auxins are discovered. So I will show you some of the slides back, which will be kind of a recapitulations for you. These auxins, most of the work has been done on the coleoptile of grasses. So coleoptile bends towards light. The very early experiments in late 1800s showed that light is perceived at the tip of the coleoptile. However, it responds at the base of the coleoptile. The base bends towards the light. And by later on, those coleoptile tips were used and the cut tips were placed on the agar blocks. These agar blocks collected auxins which are made in the coleoptile tips, and those agar blocks now can be placed on the new or another coleoptile and show the phototropic response even in the light which is uniformly present. So I will just show you 1 or 2 slides as a recapitulation of my last class. So you can see some of them, like here I will show you some of the slides where the people have done experimentally to show the coleoptile bends towards light. And then this using, so this is the experiment which I was talking to you, that coleoptile is bending towards the light. And then these tips of the coleoptile were removed and was placed in agar block or butter or somewhere a block which does not allow to pass. Then it was noticed that the agar actually can collect auxin from the tip and effect can be seen. Now, by using that collected auxin from the tips, the auxin was identified. The major plant auxin is indole 3 acetic acid. So, this much I have told you in my previous class, that auxins, by the classical studies, auxins were identified from the plant. Now, we will discuss today what we have learned in progressed from in last say 70 to 90 years. These auxins were identified as quite early around 1930 1935, and then for last say 70 to 80 years a lot of work has been carried out to understand how auxin biosynthesis takes place, how auxin signaling takes place, and how auxin transports. So, these are the three basic components of auxin metabolism that I will discuss today. First, I will talk about auxin biosynthesis, next auxin transport, and then auxin signaling. Now, coming to, as I have discussed about the difference between the classical experiment versus modern experiment, we also should appreciate the tools or technologies that have come up in last few decades which promoted auxin research. Like for example, auxins, like any other hormones, are present in a very small quantity. So, quantitating auxin actually remained challenge, and now there are techniques where you can have enhancement of the auxin amount signaling. Like for example, today it is known exactly, like for some plants at least, in a root auxin mapping has been carried out. So, each and every cell in a given root, how much auxin it contains has been measured. So, measurement technologies have improved. Similarly, there are technologies by which auxin detection is possible, like using of a promoter, using a dye or using a tagged colored molecule which would respond in the presence of auxin. So, those molecular tools I will discuss along with my classes today. Okay. So, these are the things which I have told you in the previous class, that what classical studies told us, like the chemical structure of auxins and other functions. Now, today's 21st century we will learn about auxin homeostasis, tools in auxin research, auxin action in whole plant process, interacts with other signals. Now, let us look about some tools like mutants. You can get many different mutants in different plants where you can study auxin biosynthesis and signaling, quantification of the tissue, expression of auxin inducible genes, in situ detection of auxin specific antibodies by auxin specific antibodies, and orientation of the auxin flow. So, I will discuss about those things. Now, look at this on interesting study over here. Here is a small leaf which is mentioned as label as A, and this is a leaf B, and part of the leaves are numbered from 1 to 8. So, 1 is the tip of the leaf, 2, 3, 4 and 8 and so on. As I told you, the auxin quantification has been improved. Now, even from a part of the leaves, now auxin can be quantified with GCMS. And then if you look at the auxin quantity, what is found that in a small leaf, if you go from the tip to base, there is enhancement in the auxin. That means, auxin is more in the base and less in the tip. However, if you take a matured leaf, there is no difference. So, these are of all matured leaves, there is no difference. So, this is an advancement where one can study due to the presence of advanced tools. This is another technique by which you can quantitate auxin, like you can have some kind of fluorescent protein made. Actually, in this experiment it is GFP, which gives you a color. Now, there is a machine called FACS, where one can separate out the green cells versus non-green cells. The green are the ones which are making the green protein in GFP. So, by that now one can identify the number of cells in a tissue which is expressing auxin and versus the others which are not making auxin. As I told you, there is a cell specific auxin measurement. So, auxins are measured and each and every cell how much auxin is there, one now can tell. So, these are the tools which are available today for doing auxin research. Now, this is another interesting tool. Here this allows you to monitor or measure how much amount of auxin signaling is going on. This tool can easily be understood by little bit of knowledge with molecular biology, like something there is called promoter of a gene. Now, the promoter of a gene often defines what should be the expression profile. So, here the gene which is taken is GUS or GFP. This GUS can be stained, which gives a blue color, and GFP gives green color. Now, if you have this gene under a promoter sequences which responds to auxin, then it will show the color only when auxin is there. So, there is one promoter called DR5, that is a synthetic promoter which responds to auxin. You can see over here, like some cells are showing the GUS color, but not others, which indicates the tissues where auxin is more. So, this is the, like you can see here, this is the DR5 GUS in the root tips. So, this is root and stained for the presence of the auxin, which is indicated by blue color. Now, you see in the root auxin is accumulated only in a small area which is takes like cushion center, QC, cushion center and columella and some amount in the root cap. So, root cap little bit on the cap end and columella and cushion center. So, these are the small cells where you have large number of auxin. So, you can see it again over here, this is enlarged here. So, this is the where maximum amount of auxin, and just if you go a few cells side, very little auxin. The same thing you can see when you have transgenic plant with GFP. You see the green fluorescent color is present only in the cushion center and columella. So, these are the molecular tools which is available at today or in last few decades that helped scientists to do research on where auxin is accumulated, how it is being transported and how it functions. So, another tool which also have come up, which is very important tool, is to study its transporters. So, there are many transporters. I will speak to you today about auxin transport also. You will see that how GFP protein helps in understanding how auxin is transported. Another tool which is used is also like antibodies. Antibody can be used which can bind with the auxins, and you can see this is a antibody binding, this is the DR5 GUS binding. So, this is a root. In the root tip you can see here lateral root tip, similarly you can see the antigen antibody binding over there. So, there are many techniques through which auxin accumulation can be studied. So, now we will speak about auxin biosynthesis. Auxin biosynthesis predominantly takes place in the tip areas of the shoot growing shoot tip. I told you also sometimes in one of my developmental biology classes. However, it can be synthesized in many other cells in lower quantities. Now, this auxin biosynthesis pathway has been established from 1960 onwards and has been shown that it is a derivative of tryptophan amino acids. So, tryptophan amino acids many different pathways which converts into auxin. So, you can see this pathways there is a paper in 1995 in plant physiology which has elaborated quite a few pathways. It is a tryptophan going to indole 3 acetamide acid and then 3-indole 3 acetic acid, and also it can be formed by indole 3 pyruvic acids. So, this work was published in 1995 which had the many elucidation of many several pathways. Now, today there are many genes identified, mutants identified which affect this photosynthesis, this pathway. You can see here this is the similar slide where structure is not there, but you can see that same tryptophan, tryptamine, indole 3 acetaldehyde, indole 3 pyruvic acid, indole 3 acetamide, and with the help of these intermediates tryptophan converts into auxin. There is one pathway also present which is called tryptophan independent pathway, by which indole can directly convert into IAA, the major pathway of the auxin biosynthesis. And now, as I was telling you that there are many genes identified, and here I am showing you from a paper that shows again the same pathway, but I have highlighted one area which is written as UC. This is a short form of a set of genes called UCA. And now I will show you this photograph. You look at here. This UCA, there are many UCA like genes are there, and if you knock down 1 or 2 UCA genes, you will not get a phenotype because this gene is present in multiple copies. However, if you knock down quite a few of them, this experiment they have done is they have knocked down 4 different UCA genes. In the left panel, in the panel A, you can see these are the mutants of UC1, UC4, UC10 and UC11. So, when all the 4 genes are knocked down, auxin biosynthesis is greatly reduced, and auxin is required for root development. So, you can see this quadruple mutant do not develop proper root structure like wild type does. This is the wild type root which develops very well, but not in the UCA mutant because they lack auxin biosynthesis. Similarly, auxin is also required for vasculature development. So, this is in the panel B here you can see a wild type plant Arabidopsis, and these are the vasculatures of Arabidopsis leaves. If you look below this panel C here, now you see these are the 2 cotyledonary leaves of UCA mutants. These 4 UCA mutants, quadruple UCA mutants, you can see there is no clear vasculatures. So, that also tell you that auxin is so important for root development as well as for vasculature development. So, now we will discuss about auxin transport, which is a very important aspect of auxin metabolism, and this auxin transport there are large number of research papers which talked and discussed the importance of auxins. Even people refer it is not the quantity of auxin that affects development, it is more importantly how auxin locally gets accumulated in a plant in contrast to the global accumulation or differential distribution of auxin. Now, when we discussed about auxin flow, we should talk about 2 different kind of auxin movement. There are auxin movement broadly divided into two types. One is called as a long distance transport, that is like a highway transport. One is called cell to cell communication or a local transport. The high distance transport or long highway movement takes place via vasculature. So, like from the tip of the plant, when say like in leaves auxins are synthesized, they are loaded in the vasculature. So, that vasculature connects the entire phloem. So, along with the phloem sap, it can travel throughout the plant, and that movement of auxin is irrespective as actually with respect to the phloem flow. So, auxin cannot control the direction, it is regulated by the way phloem is moving. So, it is also called as a passive flow, but more popularly this flow of auxin is called as a non-polar movement. So, pole means we understand the word by pole is a direction. So, there is a one direction which is called as a polar direction, polar movement. Now, if I talk about non-polar movement which is via phloem, it is not a directionless movement, it has a direction, but the direction is mediated via phloem movement. So, wherever the phloem goes, auxin goes by that. But the more importantly, which is important for development and formation of organs, formation of polarity, is a short range movement. That means say one cell is making auxin and how it is going to very next cell is very important, and those local movement actually make locally, say in a developing organ, locally auxin can accumulate in a small part and that can influence development of an organ. And since this movement is a directional movement from one cell to another which is assisted by the transporter of auxins, these are called polar transport of auxin. So, broadly there are two types of auxin transports. So, I will show you both the types of auxin transport and how it really affects plants growth and development. So, again now I am looking, just look at this picture I have shown you earlier. So, this is a coleoptile. This coleoptile is bending towards the light. Now, when it bends towards the light, has been shown that this part which is outer side of the bending cells are bigger and the inner side cells are shorter. So, you can say also otherwise, there is elongation took place more in the side which is away from the light. So, the cotyledon bent. Now, if you take out these cells from this side and this side, if you look at the auxin concentration, you will find that there is more auxin in this side than this side. So, you can infer in a way that auxin have moved from the lightened area to the shaded area. So, auxin can move and this movement is a polar movement. This is not assisted by vasculature movement because there is no vasculature which connect from one side of the radial side to the other radial side directly. So, these are polar movement. Now, similarly you can look at this slide here. So, this one was by auxin concentration and here by DR5 gas. I told you earlier just a few minutes back that DR5 is a synthetic promoter and that responds to auxin and gas gives the blue color. So, wherever auxin is more, there will be more green color. So, this seedling here is bending towards the light. So, this is the source of light, seedling is bending. Now, if you take the bend area and do a gas staining, you will find this side which is outer side of the bend has more auxin than the inner side. In other words, auxins from this area which is lighted area has moved towards the shaded area and so the auxin is much more over here. auxin also play very important role in gravitropism. So, this also I told you in the previous class that auxin has a very important role in gravity determination and growth of. If you take a seedling like this, if you just place it horizontally, what you will find is that root bends towards gravity and the shoot end goes away of the gravity or goes upward. Now, this becomes a very interesting question, how auxins behave. Do the auxin behave separately? Is the auxin from here going upwards and here auxin is coming downwards? So, what happens? And that kind of experiments has been recently carried out with the modern technical tools. Now, you see here, this is a coleoptile which is growing straight, growing vertically, and over a few days it has grown bigger. So, these are the cells of this area has gone big, and here you can see how cell elongation is taking place in the coleoptile. Now, if you place same coleoptile horizontally and then take at the length of the cell, what you will find that as the time passes, the upper side, you can see here, this upper side growth is like this. So, there is a very little growth in the upper side, however, lower side of the coleoptile length, it has gone into bigger length, the cells have gone bigger. So, as exactly what I told you in the previous case, here also in response to gravity, the cells of the downside of the shoot tip or coleoptile goes longer. So, it goes up. And now, if you again look at the auxin level, you will find that the lower side has higher auxin than the upper side. However, if you grow vertically like this, if you grow vertically, these two sides have almost similar level of auxin, but if you grow horizontally, there is more auxin in the down. That indicates you that the auxin from upper end goes down, reaches down towards the cells of the lower side. Now, again look at the root here. The previous one was shoot and this is an experiment with root. Again the DR5 gas, what you can see, if you put it horizontally, within a few hours the auxins accumulates to the lower side. So, it does not matter whether it is root or shoot, auxins come to the lower side. Now, again the interesting question comes, so how it differentially respond, because a seedling is placed horizontally, auxin is more in the root, auxin is also more in the shoot to the lower side. Now, the answer lies in one of the graph that I showed you earlier in the previous class. The root and shoot differentially respond towards the auxin concentration. If the concentration of auxin goes high beyond certain level, it inhibits root growth. So, here when auxin concentration is very high, the cells will not grow, but it will grow more in the upper side. However, in case of stem, the stem cells can tolerate much higher level of auxins. So, up to this much of auxin, stem will have a positive growth. Only beyond that auxin, stem will have a negative growth. So, when a seedling is placed horizontally, the higher level of auxins in the stem end promotes stem growth positively. So, lower side grows more and it bends upward. In root, the opposite thing happens. So, all of them are important determine how the auxins movement affect growth of a plant. Now, these are the auxin pathway which I told you. This is one thing is long distance flow through phloem and short distance through cell to cell. Now, look at this structure here. This is a plant and here the central column is a vascular column. In the vascular column, auxin is flowing from the shoot tip all the way to the root end, which is cushion center. And this movement you will call it as a non-polar movement, because this movement is carried out by the effect of phloem flow. Now, in contrast, you can see a flow which is mediated by cell to cell. This is a group of cell of a tissue and the arrow indicates the flow of auxin in the tissue. And this flow of auxin is determined by a class of transporter which are called as PIN, P-I-N, PIN. This PIN class of transporters arrangement will tell which direction auxin will flow, and here it is marked by this yellow color. Like this yellow color marking in these cells indicate the auxin will come this way or down, but it will not go other way around. So, the transporters which is present in the side will determine which direction auxin should flow. This is the brief about auxin transport in the plant, and I think we should understand little bit about before we go for the next part of it. See, this auxins is indole acetic acid, IAAA. This indole acetic acid IAA, which is in the cell, can take up a hydrogen ion which can form IAAH. Now, this auxin, suppose this is a cell, one cell, this is next cell and this is the other cell, and you have the spaces between the cells. The spaces between the cells are called apoplast. These auxins are present both inside the cell, in the apoplast, and then is moved to the next cell. Now, what happens is very interesting thing about the pH of the cell. So, pH of the cytoplasm is roughly around 7 to 7.3. However, pH in the apoplast, which is between the cells, is quite low, where pH is right about 5.8, roughly around 5.8. So, what happens is that since pH is low, it is more acidic, it has more hydrogen ion. So, IAA, indole acetic acid, which is normally negatively charged in neutral pH, becomes protonated and becomes IAAH. So, when it becomes IAAH form, it loses its charge and it becomes more of a nonpolar molecule. And when it becomes a nonpolar molecule, it can pass it through the hydrophobic plastid layer, plasma membrane layer. If you recall, the plasma membrane layer is mainly a hydrophobic layer in the core of the in the both the sides of the bilayer, the hydrophobic. Now, this hydrophobic bilayer, when auxin is protonated, it is nonpolar, it can pass it through the lipid bilayer, but once it comes inside the cell, it loses the proton, because inside it is a neutral pH around 7.3 and it becomes charged molecule. So, charged molecule indole acetate, it cannot pass it through the lipid bilayer, so it needs transporters. So, there are a few kind of transporters are there in for auxins. One of them group of transporters are called PIN, PIN. The name came because many of such transporters, when they are not present, they do not form any leaf. So, the stem looks like a bear, like a tip of a pin. So, and these pin proteins, there are many of them in Arabidopsis, there are at least 7 of them which play a role not redundantly, in the sense it is not that all of them are non essential, all of them are not redundant, they express at different time point and they do different jobs. And those pin proteins can carry auxin from one part of the cell to the other part of the from one cell, and it can throw out to the next cell apoplast. So, I will show you that in a minute in one of the slides here. Okay, so this slide, yeah, right. So, this is the auxin transport system. You can see here these auxins can transport via pin protein and PGP protein. It can go to the next cell. So, arrangement of the pin, like if the pin is this side, auxin will move towards right. If pin is down, auxin will be thrown to the down, and so on. So, the next cell auxin can enter. However, auxin which is also protonated auxin apoplast can get an auxin carrier through which it can enter to the cell. So, entering of the cell is dependent on the carrier, passively carried out, but exiting a cell requires active transporter which are like pin proteins. So, in brief, this is something like this: pH 5, pH 7, IAH enters, and then it cannot go out because it is already charged, and then it takes the help of these transporters. All the transporters are usually arranged in one side. So, auxins, this will be a neat flow of auxin. So, if you take a few cells like this, what you will see, the auxin will go from the left cell to the intermediate place, and then from this cell to the right side. So, auxin will have a neat flow from left to right. So, this is how auxin flows. Now, the pin protein can be visualized if you can hook with a protein like GFP. This GFP emits green light. So, if you hook up with pin, then wherever pin expresses, you will find the green color is expressed. You can see in this case, here this is a cell where pin 1 promoter and pin protein fused with the GFP. So, it will make pin GFP, and that will be colored arranged in the plasma membrane. So, you can visualize like this. So, this is one of the picture where pin GFPs can be seen, and you can see these are the green colored which are arranged in the plasma membrane of the cells which are in tip of the shoots. Okay. This pin protein can relocalize. Say suppose one time pin is in this side, it can recycle, can go to endosome and can reorient in the other side. So, once the auxin is flowing outwards towards upwards, it can now later on can flow downwards. So, the way auxin will flow will be defined by the way pins are arranged in a cell. These slides I have shown you probably earlier, and this shows how auxins flow from one cell to other, and eventually in an embryo, this is an embryo. So, this is only small embryo, single cell embryo goes big and it forms a heart shaped embryo, and then you will find auxin is one part of the cotyledon, in another tip of the cotyledon, and more in the down where root will form. So, auxin will be present in this hypophysis part, tip of the cotyledon, but very little in this area where shoot apical meristem comes, and pin proteins play a very important role in determining where and how much auxin will be accumulated. This is a beautiful picture where you can see how pin GFP gets relocalized. This is a very early embryo, pin is throughout even early globular stage, but in the late globular stage you can see how the pin is localized in lower cells. So, the distribution of pin protein will determine where the auxin will be maximum, and this is the places where auxins are all brought from the globular embryo to the hypophysis area, and this is the mature one. Okay. So, earlier I showed you one mutant of UCAs. So, there are 4 UCA mutants where there was no root. Similarly, if you have a alteration in the pin1 function, like here is in a mutant called GN mutant, which is required for the distribution of the pin, what you will find that they are also defective, there is no root formation. This is the wild type seedling and this is non-mutant seedling. So, there is no proper root formation on this mutant. So, similar to biosynthesis mutant, distribution mutant also have a very similar effect. Now, coming to the last part of my discussion today, which is auxin signaling. This is very important, but most of the students who are taking botany, plant physiology or any kind of plant courses in your undergraduate chapter, you must be learning this in your books. Auxin signaling has been thoroughly characterized, and the auxin signaling mechanism is to a certain extent is similar to many other animal hormone mechanism and many plant hormone mechanism. Like here what happens is that auxin's major effect comes in terms of regulation of transcription. So, I told you also you probably you know about it, transcription of a gene is regulated by its promoter, and the promoter needs transcription factors. In case of auxin, there is a transcription factor which are called ARFs, auxin response factor. These ARF transcription factors bind with the promoter of auxin response genes and carry out auxin signaling. Now, what happens in normal cell where auxin is not there, the auxin response factor is inhibited by a repressor protein. This repressor and transcription factor both combinedly bind with the promoter, and because of the repressor the responsive genes cannot be transcribed. So, once auxin comes, it helps to remove or degrade the repressor, so that now auxin response factor can function, and that is how the auxin signaling goes on. So, I will help you to understand this auxin signaling with some of the cartoons and some of the experimental data which are available. So, in brief, if you look into this, there are 2 aspects which I told you. In one aspect is the auxin biosynthesis, second is the transport, and then importantly it has to be perceived. So, there must be a receptor which will receive auxin, and this will activate a transcription factor which will activate in turn the defense related, so the genes which are target of the auxins and its biological functions. Now, in terms of auxin's effect by the repressor, by the mode of the repressor, auxin signaling is broadly classified into 2 groups. In the top I mentioned is SCF-TIRNR1. The name sounds quite big, but this is the most commonly understood and described in the books on the literature. This is the SCF-TIR1, this is a receptor. Receptor can bind with the auxin and can activate auxin signaling. There is another receptor which is ABP1 that also can activate signaling, but the function is a little bit different. The first one, SCF-TIR1, when it binds with the auxin, that dictate development and differentiation, whereas binding with ABP1 is important for cell elongation. So, if you now look at this aspect, I will tell you a little bit of development and differentiation. See, this is the cartoon which I was trying to describe to you. This is a auxin responsive gene, and there are thousands of such genes which are induced by auxins, and this promoter has the binding site for auxin response factor or ARFs. These auxin response factors potentially should be able to express auxin responsive genes, provided it is not inhibited by AUX or IAA. This is a protein which is named as either AUX or IAA, because these are the ones which are required for AUXAA inhibition and signaling. So, when this inhibitor molecule, this AUX inhibitor molecule binds with the ARF, then because it is a repressor, it does not allow transcription of auxin response gene. And TIR1 is the receptor of auxin. This receptor of auxin actually is a co-receptor with AUXIAA. This AUXIAA is a repressor and also co-receptor of the auxin. When this binds, this TIR1 can lead to degradation of auxin IAA. This is a one protein which can ubiquitinate cell. Actually, some of the students may know that there are several pathways by which proteins undergo degradation. One of the pathway is called ubiquitination and proteasomal degradation. This TIR1 protein helps, which is activated by auxin, helps in degradation of AUXIAA. Now, if you recall, I told you AUXIAA gene is a repressor, and when it is degraded, ARF is free, and this auxin response factor which is free now can drive auxin responsive genes, and this is how the auxin signaling takes place. So, you can also see from here that unlike many different kind of signaling which students might have read, like auxin signaling, like calcium signaling, G protein couple receptor signaling, light signaling, unlike those, this signaling pathway is very short. So, there is only one single step inhibition and then directly activate transcription factors, and there is no intermediate MAP kinase signaling which is present between auxin and transcription of the genes. So, now you can see here three plants. This is wild type, here ARF is present, and when auxin comes, this AUXIAA is degraded. So, auxin functions. So, you have both shoot and root. This is a mutation in the ARF5. ARF5 is a auxin response factor, this is a transcription factor. In this case, the transcription factor is not there, ARF is not there, but repressor is there. Since transcription factor is not there, it does not transcribe. So, auxin response does not work and there is no root formation in the mutant. Similarly, if you look in a case where the AUX or IAA could not be degraded, here auxin is there, but due to some change, it is called gain of function mutation, where this IAA is not being able to degrade. So, auxin is there, ARF is there, everything is there, but only this is a mutant IAA which is resistant to degradation, and then there is no signaling. Look at the phenotype, both the case it is same, there is no root. And also I showed you this similar phenotype by in the UCA mutant. So, auxin biosynthesis defect auxin's response factor mutant defect or defective in auxin repressor binding has a very similar phenotype, which is a rootless phenotype. Now, this is coming to the almost last part of my class today, is the another receptor which I was telling you, ABP1. This part of the auxin signaling has very lately discovered, but the effect were known at the earliest time of auxin discovery. Like I told you, auxin helps in cell elongation, by which actually through which auxin was discovered. So, this TIR on SCF where has been mostly responsible, has been shown to be responsible for development and differentiation. The reception by ABP1 is responsible for cell elongation, and I will speak to you a bit about it. Now, this is a cartoon where you can see this is a plasma membrane cell of the cell, and there is a receptor ABP1. This ABP1 protein is synthesized in ear, get transported and present in the plasma membrane. Now, when auxin binds, this helps in proton pump activation. So, there is a proton pump which gets activated. So, ABP1 can activate a proton pump. So, more hydrogen ion is pumped towards the outside, and there is some problem. Okay. So, that is okay. So, whenever the proton is more pumped outside, it acidifies, and that can activate a set of proteins which are like expansion proteins, and which eventually work on the cell wall. So, eventually the cell wall loosens, cell elongates, and that is how the elongation of the cell takes place. Now, that has also been shown, was known in the classical studies, that when you apply auxin, it affects in acidification of the wall. So, there are two these events which we should be understanding or remembering separately. One role of auxin which is mostly affecting at the transcriptional level, which regulates large number of gene expression via activation of auxin response factor. The default function of auxin response factor is transcription, but it is inhibited by the repressor. The repressors do not allow it, and auxins degrade it, and eventually that helps in differentiation process and developmental process. The other aspect of auxin, what I told you, is auxin can activate cell wall elongation, and in fact the experiments have been proven that if you artificially acidify a wall, like if you do not apply auxin at all, but artificially acidify one small part of the wall, and then there will be elongation of that part of the cell wall. So, it may be nothing much to do with the transcriptional regulation of cell wall elongation, it may be much more related the activation of the proton pump. So, now in brief, and so these are the two classes which I have completed for you on plant hormone. So, basically today if you today and yesterday's class on auxin, I have told you how auxin is important, and the totally it looks like auxin is indispensable for survival of the plant. From the very single cell stage, when the life of a plant began, when there is only single cell, auxin starts transporting from one cell to the other, lower cell to the upper cell, and then as and when more and more cells grow, the life become bigger and bigger by a few number of cells, auxin starts taking a very pivotal role, and auxins move at different points in a different concentration. Usually in a growing embryo there are 3 poles where auxins transports, and one pole where auxin is maximum, it makes a root. And if you do not have enough auxins, there is no root development and plants do not survive at all. Besides the root development, I have told you also vasculature development, the transport system like your blood vessels, which carries the food, nutrients, metabolic signals among the tissues in the system. The vasculature system development absolutely depends on auxin. So, if there is no auxin, there will be no vasculature development. And then also I told you that auxins, how it is so important for the cell elongation. Auxin is very, very important for initiation of the leaves, like this is where the pin mutant came up. In shoot apical meristem, in certain points auxin accumulates. Wherever the auxin accumulates, there will be leaf. So, if auxin accumulation is blocked by a pharmacological experiment, like you apply an auxin inhibitor, then leaf do not form. You have a mutant of a transporter where auxin cannot move from one side to the other side, then there is no leaf formation. So, auxin movement is very, very important, and differential amount of auxin accumulation will dictate where a leaf will form. This will again continue in the leaf serration. Many of the nature leaf margins are serrated. There are many compound leaves formed. Sometimes one leaf are simple, sometimes leaves are compound. So, wherever the compoundedness is required, there the differential auxin is required. Wherever leaf must be wrinkled, more cell has to be divided, more cell should continue to grow for longer period of time, auxin play a very important role. So, in summary, I should tell that auxin is an indispensable hormone. Without auxin, life does not begin of a plant, life cannot continue and cannot sustain. So, auxin is very, very important hormone, and thank you so much.
With this note, thank you sir, thank you so very much for giving us a very, very prolific session. Dear friends, I would like to tell you all that if you have any queries or if you want to give your feedback regarding this particular lecture, then you can mail us at info.cc@nic.in. We would love to solve your queries the next time when Dr. Ashish Nandi visits our studio. And dear friends, keep watching us with the help of YouTube, as very soon we are going to upload this lecture on YouTube, with the help of which you can access this lecture as well as other lectures in continuation on plant physiology. So, we will be meeting again very soon and would be talking to you with the help of this EduChat platform. Take care, goodbye. Thank you sir, thank you so very much.